Hello everybody. So today we are going to discuss cell physiology. Under cell physiology, we are going to discuss three main topics. Number one, cell membrane. Okay, commonly known as plasma membrane. Number two topic we are going to discuss is cell cytoskeleton. Okay, and number three that we are going to discuss is cell-cell interaction. Cell-cell interaction, which is also known as intercellular connection, right? So this topic we are going to discuss today. So first topic is the cell membrane. What are the important things we have to remember for our MCQ purpose? So, cell membrane, all of you know that it is also known as the plasma membrane. First, you have to remember the thickness of plasma membrane or cell membrane is on an average 7.5 nanometer, right? Then second thing we have to remember that plasma membrane is a lipid bilayer. We all know this thing. So if you look at this diagram, okay, so you will be finding out here that this is one lipid layer and this is another lipid layer. If you look at this lipid layer, I will simplify this diagram. What you are finding out one polar head or hydrophilic head along with two hydrophobic chain. So this is typical structure of phospholipid, right? So this is one layer of phospholipid. And you are finding out another layer of phospholipid is arranged in opposite direction. So this is the polar head of the phospholipid and these two are the hydrophobic chain. So now many of these phospholipid molecule will be arranged like this, okay? So that the polar head faces the surface and hydrophobic side chain they faces at the interior of the membrane. So we can understand easily that this surface of the membrane, both the surface of the membrane contain the polar head of the phospholipid molecule, hydrophilic head of the phospholipid molecule, and the interior of the membrane is mainly of hydrophobic region of phospholipid, right? So both the surface is hydrophilic, okay? One of the surface will remain in contact with the extracellular fluid and another surface will remain in contact with the intracellular fluid, and interior of the membrane contain hydrophobic region. And inside this there will be multiple protein molecule like this. So in between there will be multiple protein molecule like this. So these proteins are different types. We will be discussing this. So basically we can understand that the plasma membrane is mainly composed of lipid bilayer. So this is one layer of lipid, this is another layer of lipid, and the proteins are embedded in this lipid bilayer. So if we look at the composition of the plasma membrane, so we are writing here the composition of plasma membrane, important topic for MCQ. So number one is lipid, right? So lipid, according to the mass or according to the weight, the percentage of lipid is 45%, okay, 45%. So what lipid is most commonly present? The phospholipid is the most common lipid in the plasma membrane, followed by cholesterol. So this information you have to remember, that phospholipid is the maximum lipid which is present in the plasma membrane, followed by cholesterol. And the lipid which is not present in the plasma membrane is the triglyceride. The percentage is the 0%. This is also an MCQ. triglyceride is not present in the plasma membrane. Now, if the question is asked which kind of phospholipid is predominant in membrane, then your answer will be phosphatidylcholine. Okay? Phosphatidylcholine, CHO, right? So this is the most common phospholipid which is present in the membrane. Now we all know that phospholipid is the amphipathic lipid that we have already discussed, that one region will be the hydrophilic region and the hydrophobic tail will be there. So this is the amphipathic lipid which is present in the membrane. The second composition that we have to remember is protein. Okay, protein. Percentage is 55%. So make it clear that although we know that plasma membrane is a lipid bilayer, but if you look at the percentage according to their mass, then protein is a little bit more than lipid in most of the biological membrane, and the percentage is 55%. Now, according to the location and properties of the protein, there are two types of protein I have to remember. One is known as integral membrane protein, which is also known as intrinsic membrane protein, integral membrane protein or intrinsic membrane protein. And second one is known as peripheral membrane protein or extrinsic membrane protein, right? So integral or intrinsic or peripheral or extrinsic type of type of membrane protein, right? So now we will look into this diagram. So look at this diagram. So what you are finding out? You are finding out that this is the lipid bilayer. So this is the lipid bilayer, right? We have already discussed. And inside this lipid bilayer, we are finding out multiple protein. So this is one protein which is present through and through the cell membrane, means it is spanning the cell membrane, present through and through. It is spanning the membrane. Okay. Similarly, this is another protein which is also spanning the membrane, but this protein is basically a channel. So basically, membrane protein means they can be of channels, they can be of transporter, they can be of pump, anything. But if we look at the property and location of the membrane, some of the proteins are present through and through the membrane that span the membrane through and through. That kind of protein is known as integral protein. They are very difficult to separate from the membrane. That's why they are known as the integral part, intrinsic part of the membrane, right? They are very difficult to be separated from lipid bilayer. This kind of protein is known as integral protein, so present through and through. We have to remember that integral protein remain attached with lipid bilayer by which kind of forces? Your answer will be hydrophobic interaction as well as hydrophobic interaction as well as van der Waals forces, right? So this is very, very important, that integral membrane protein with they remain attached with the lipid bilayer by hydrophobic interaction and van der Waals interaction. PGI Chandigarh MCQ that has already been asked here, okay? So please be careful. And the second kind of protein is the peripheral protein. Look at the diagram once again, right? So now if you concentrate on your integral protein, so this is one integral protein, right? So I can find out along with integral protein, another protein is attached, right? Another protein is attached, and this protein is known as peripheral membrane protein. So first thing is that peripheral membrane protein means they will remain attached with integral membrane protein, or some of the peripheral protein they may directly attached on one of the surface of the membrane, maybe in the inner leaflet of the membrane or maybe on the outer leaflet of the membrane. That means if you look at this protein, if you look at this protein, if you concentrate on this protein, you will be finding out that this protein is only attached on one surface of the membrane, but it is not spanning the membrane through and through. So this kind of protein is also known as peripheral or extrinsic protein. So what is the bottom line? Bottom line is that peripheral membrane protein, they may remain attached with integral membrane protein also, or they may remain attached with directly with membrane also, maybe in the inner leaflet, maybe on the outer leaflet. This peripheral membrane protein which remain attached with integral membrane protein, the forces we again have to remember, right? So here the forces is electrostatic forces, as well as hydrogen bond. Okay, so this kind of forces I have to remember. And the peripheral membrane protein which remain attached directly with the membrane. Okay, the forces here is covalent interaction. The forces here is covalent interaction. So please remember this information, that the peripheral membrane protein which remain attached with integral one, they attached with electrostatic forces or hydrogen bond, and the peripheral membrane protein with directly attached on the membrane, they remain attached with the help of covalent interaction. This is very important. So this is all about the protein component of the membrane, right? And the third composition of the membrane is carbohydrate. Although the percentage is very less, 1 to 3% only, but they are also present in the form of glycoprotein or glycolipid. So this is the whole composition of the plasma membrane: lipid, protein and carbohydrate. Now I already told you that the lipid percentage is 45% and the protein percentage is 55%. Means in biological membrane, if you look at the protein, okay, is to lipid ratio. Protein is to lipid ratio. Okay. Almost they are equal to one another. 1:1. Although I am writing here that 45% and 55%, but they are almost same to same. So grossly we take the ratio that the protein and lipid is to 1:1. Okay. But this ratio is not 1:1 for every biological membrane. Okay. There are certain biological membrane where protein is very high. So what will happen? The ratio will be also very higher. So there will be an MCQ that this protein is to lipid ratio. So this ratio is maximum in which biological membrane? Right? Could you guess? Your answer will be inner mitochondrial. Inner mitochondrial membrane. Why inner mitochondrial membrane? Because we know the inner mitochondrial membrane contain the proteins, all the proteins of electron transport chains, and that's why if you take the protein is to lipid ratio, the ratio is maximum, and that is 3.2. So protein is to lipid ratio is 3. Second is sarcoplasmic reticulum. The sarcoplasmic reticulum, the calcium storage organelle, all of you know, it also contain huge number of pump, huge number of channels. That's why the protein content of the sarcoplasmic reticulum membrane is also higher, and that's why the protein is to lipid ratio is 2 here in case of sarcoplasmic reticulum membrane. So obviously, if I am asking that the ratio is maximum for which biological membrane, right? So question can also be given that the ratio is minimum, that this ratio is minimum in which biological membrane. Minimum means you have to search one membrane where the lipid is very high in comparison to protein. Then and then only the ratio will be minimum. So all of you know that the myelin sheath, okay, myelin sheath, okay, contain huge amount of lipid. Basically, we all know the myelin sheath is a insulated covering on the neuron, which is nothing but the mainly the plasma membrane wrapping up the neuron. So that's why it contains huge amount of lipid, and the ratio between protein to lipid is only 0.23. So these are the all protein, lipid, and composition of the membrane that you have to remember for your MCQ purpose. Now the next thing is that in the plasma membrane, okay, the recent concept of plasma membrane is that it's a lipid bilayer which forms a gel-like appearance, right? So the recent concept of plasma membrane was given by two scientists. They are known as the Singer and Nicholson. What is the concept? Concept is that the lipid bilayer form a gel-like area. The lipid bilayer form a gel-like region. So this is the gel, lipid bilayer. And on this gel, there are multiple protein embedded, and these proteins are like icebergs. So on our gel, there are multiple icebergs. This protein or the iceberg, they can move from one to another direction. This is known as the lateral movement of the protein inside the lipid membrane. Okay, that means we can understand that the proteins which are embedded in the membrane, they are not the fixed structure. They are moving from one part to the other part of the membrane like an iceberg. And if you take a view from above, right, how it will looks like? It will looks like a thick gel. Onto that some protein, and it will give you a mosaic appearance. That's why this model is known as fluid mosaic model of plasma membrane, which was first time proposed by Singer and Nicholson. So gel or fluid phase, and on this gel some proteins are moving. It is giving an appearance of mosaic pattern. So that's why this is known as fluid mosaic model of membrane, right? So just now I told that the lipids, they forms a gel like area, right? So if I increase the temperature of this membrane, what will happen, right? So this gel will be converted into liquid phase. So the temperature at which the gel, okay, the lipid bilayer, the gel is converted into fluid phase. Okay, so if you increase the temperature of the membrane, the lipid bilayer which forms a gel, that will be converted into fluid phase. This conversion, okay, this conversion, suppose it is occurring at a particular temperature, that temperature is known as transition temperature or TM of the biological membrane. Transition temperature means the temperature at which the plasma membrane is converted from gel to fluid state, that is known as the transition temperature. Now the transition temperature depends on the composition of the membrane. If the plasma membrane contains huge amount of saturated fatty acid, okay, if it contains huge amount of saturated fatty acid, more the saturated fatty acid content of the membrane, more will be the transition temperature. Means you require more temperature to make this gel to liquid, okay, gel to fluid, right? That means more the saturated lipid means the membrane is becoming more harder, okay, it is very difficult to convert it from gel to solution phase or gel to liquid phase. right? Now, there is an inherent question in this topic that what is the role of cholesterol in this transition temperature, right? So please remember that what is the role of cholesterol. So here you have to remember that if the given temperature, okay, if the given temperature is below the transition temperature of the membrane, then cholesterol, okay, increases the fluidity of the membrane. Cholesterol increases the fluidity of the membrane. Means when your given temperature is below the transition temperature of the membrane, more the cholesterol, more will be fluidic. The membrane will be more fluidic. But when the given temperature is above the transition temperature of the membrane, in these cases, the opposite thing will happen. That means more the cholesterol, more will be the decrease in fluidity of the membrane. Means above transition temperature, cholesterol decreases the fluidity of the membrane. As means we can remember that cholesterol is making the membrane harder when the temperature is above the transition temperature. So this is all about few points, important points about plasma membrane that you have to remember for your MCQ purpose, right? So now we are moving on to the next topic. So our next topic is cell cytoskeleton, right? Cell cytoskeleton. So cell cytoskeleton means it is like our bony skeleton, right? The particular shape of our body, the upright posture of our body is maintained by bony skeleton in our body. Similarly, if I ask you how a cell maintain its shape and size, your answer will be there are presence of cell cytoskeleton which maintain the size and shape of the cell. So the first thing is that cell cytoskeleton means they maintain the size and shape of the biological membrane, right? So what are the different cell cytoskeleton? Okay, so let's classify it. So cell cytoskeleton are of two types, okay, according to the nature. One is dynamic type of cell cytoskeleton and other one is static type of cell cytoskeleton. So dynamic and static. Okay, static means fixed. Okay, so the cytoskeleton which are static, they're the fixed cytoskeleton. Fixed means the length of this cytoskeleton is not going to change according to the cell growth. Okay, the length is not going to change according to the cell growth. But dynamic means when the cell is growing, the length of the cytoskeleton will also grow proportionally. If the cell length is decreasing, the length of the cytoskeleton will also decrease. That means dynamic cytoskeleton means the length is not fixed, and the static cytoskeleton means the length is fixed one. Now this dynamic cytoskeleton, again there are two types. Okay, very, very important. One is microtubules, okay, and second one is the microfilament, right? And static cytoskeleton, there are only one type, that is nothing but intermediate filament, right? So intermediate filament is a fixed structure inside the cell which are not going to increase or decrease according to the length of the cell, right? So that is intermediate filament. Now, if you look at the diameter of these three different cytoskeleton, then the strongest cytoskeleton is microtubule. It has a diameter of 25 nanometer. Microfilament diameter is 7 nanometer and intermediate filament has a diameter of 10 nanometer. So microtubule is the thickest or strongest cell cytoskeleton which is helpful for maintaining the cell size and shape. Also others are there, like microfilament are also there and intermediate filament are also there. Now let's try to discuss a little bit detail about all these three cytoskeleton. So look at this diagram taken from genome, right? So this green one, this is your microtubules, okay? This is your intermediate filament, a solid structure, and this is your microfilament, right? So first discuss these microtubules. So what you are finding in this diagram? You are finding in this diagram that it is composed of small globular subunit, okay, which is forming the wall of this microtubule, right? So I can understand that the wall of these microtubules is formed by subunits, and these subunits are known as tubulin subunits. Tubulin subunit, right? And there are two types of tubulins, mainly alpha and beta tubulins. So if you look at the arrangement of this microtubule, then one alpha tubulin followed by one beta tubulin, again one alpha tubulin followed by one beta tubulin. So this way 13 tubulin subunit will attach with one another and it forms the periphery, it forms the wall of the microtubule. So this is fixed, this 13 number is fixed. So that's why you have to remember, because MCQ can be given from here. So the wall of the microtubule, okay, is formed by 13 tubulins, alpha beta, this way, right? Apart from alpha beta tubulins, you also have to remember a separate kind of tubulin, that is gamma tubulin, okay? So this gamma tubulin is part of centrioles. So it is present at the level of centrosome. You know that centrosome contains centrioles, the centrioles contain the microtubules, and the microtubule which forming the centrioles, they contain the gamma tubulins. So obviously the role of this gamma tubulin is during cell division, it forms a mitotic spindle. So the gamma tubulin is forming the cell division, but the other type of tubulins, the tubulins which we are discussing here, alpha and beta tubulins, which forming the microtubule, they also has other functions, okay, other functions we'll be discussing. So what is the functions of these microtubules? So microtubule, okay, so obviously it is a part of the cell cytoskeleton, means it will maintain the cell size and shape. Apart from that, you have to remember that microtubule is present inside the cilia, okay? It is also present inside the flagella. So for the movement of the cell, this microtubule is also required. So inside the cilia, inside the flagella, this microtubule is also there. And the second thing is that microtubule is also responsible for intracellular transport process. Intracellular transport process. If I ask you how a molecule moves inside the cell from one part to other part, there is a transport process which is known as the intracellular transport process. We'll be discussing this topic in neuron chapter, right? So this intracellular transport is also based on this microtubule. Microtubule is also responsible for cell motility in the form of cilia and flagella, and obviously we have discussed that it's a part of, okay, centrioles. So that is a special type of microtubule, and obviously the microtubule is a cell cytoskeleton, means it will be helpful for maintaining cell size and shape. Okay. Now I told you the microtubule is a dynamic structure. So what do you mean by dynamic structure? I told you already that the length of this microtubule can increase or decrease according to the need of the cell. So there is a process, okay, when the cell is growing, more and more tubulin molecule, okay, will be polymerizes, okay, will polymerizes here, okay. So this is known as the polymerization of alpha and beta tubulin. Polymerization of alpha and beta tubulin. More and more polymerization means what will happen? There will be increase in length of this microtubule. So where the length of the microtubule is increasing, this is known as the growing end. Growing end of microtubule is also known as the plus end of microtubule. Please remember. So the plus end means it is the growing end. When the cell is growing, the microtubule will also grow. And that is due to polymerization of alpha and beta tubulin at the end of microtubule. Okay. So polymerization require energy, source of energy. The energy source for the polymerization of the microtubule is given by GTP, guanosine triphosphate. This can also be an MCQ because for microfilament, this is not GTP. For microfilament, this is adenosine triphosphate. But for microtubule, the source of energy is guanosine triphosphate, right? Similar to the polymerization, there may be possible that microtubule length can also be decreased. Decreased length will occur whenever more and more tubulin molecule will depolymerizes from other end of the microtubule. So depolymerization of the tubulin subunit from other end. So what will happen? The length of this end of microtubule will gradually decrease. So this is non-growing end of the microtubule. Non-growing end is also known as minus end of microtubule. Getting my point? So dynamic structure means, okay, there will be a positive end, there will be a negative end. Positive in the growing end and negative in the non-growing end. That is all about microtubule that you have to remember, right? Then second, you reach here the microfilament. Okay, this is also a dynamic structure. So microfilament, make it clear first. It is nothing but actin. Oh my God. Okay, yes, microfilament is nothing but actin. What do you mean by that, sir? So if you look at the microfilament structure, it is also composed of globular subunit, right? So this individual subunit of microfilament, they are known as the G-actin. They are known as G-actin. So this is one G-actin. This is another G-actin. So this is again a G-actin. This is another G-actin. So whenever more and more G-actin, G stands for globular. G stands for globular. Okay, so when more and more G-actin attached with one another, it forms a long filament. This long filament is known as filamentous actin or F-actin, right? Filamentous actin or F-actin. So if you look at this diagram, you will find out that there are two filamentous actin. They are wind with one another and it forms a whole microfilament. So two F-actin molecule, they wind up with one another and it forms a microfilament. So the microfilament is nothing but actin. Which actin? Filamentous actin or F-actin. Now make it clear. Muscle contraction is also based on actin myosin. Is the same actin is also present there? Answer is yes. Okay, whenever I am saying that actin myosin interaction is causing muscle contraction, that is also an actin. Which actin? F-actin. But generally we say actin means it is a filamentous actin. That's why we never say F-actin, 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 right? But it is a filamentous actin. It is an F-actin which is also causing muscle contraction. Okay, so that's why this actin or the filamentous actin, it is not only part of the cell cytoskeleton, it is also the component of the contraction of the muscle, it causes actin-myosin interaction. Okay, getting my point? So it is present in almost every cell, actin. And that's why in Genome one sentence is written that actin is the most abundant intracellular protein which is present in the mammalian cell. Actin is the most abundant intracellular protein present in mammalian cell. Okay, so because it is not only part of the cell cytoskeleton, it is also responsible for contraction of the muscles, right? So globular actin and F-actin. Again the same thing, because the actin is an dynamic cytoskeleton, what will happen? When the length of the cell is growing, more and more polymerization of the globular actin will happen, and there will be increase in length of the actin. That will be known as the plus end of microfilament, and the other end is the non-growing end, that is known as the negative end of microfilament, right? So this way you have to remember that microtubule and microfilament, they are the dynamic structure. Now look at the fixed structure inside the cell, and that is known as intermediate filament. Okay, you are not finding out any subunit here, because for a particular type of cell, particular intermediate filament is there. And in molecular biology books, it is written that there are altogether 70 different varieties of intermediate filament has been discovered from different types of cells. Okay, so the bottom line is that intermediate filament is a fixed protein inside the cell because of the fixed nature. Okay, when the cell is growing or when the cell is proliferating, the protein, the intermediate filament, will remain fixed inside the cell. Because of this property, the intermediate filament is used as a cell marker. It is used as a cell marker for identification of different types of cell. What do you mean by that? Means if I give example, it will be more clear. Suppose I am giving you example of one intermediate filament which is nothing but cytokeratin. All of you know cytokeratin is present in which cell? Okay, it is present in epithelium cell. It is present in epithelium cell, right? So it is a marker of epithelium cell. What is cytokeratin? Cytokeratin is an intermediate filament. So suppose you have given a tumor biopsy. Okay, you have to identify whether the tumor has originated from epithelial cell or the tumor had originated from mesenchymal cell. What will you do? If you stain this pathological section with the stain which is staining the cytoskeleton, and if it is positive for cytoskeleton, then you are 100% sure that the tumor has originated from epithelial cell. Similarly, the other intermediate filament is vimentin. Okay, vimentin. This is a marker of mesenchymal. It is a marker of mesenchymal cell, right? Similarly, there are other intermediate filament also, like for example desmin, right? So it is a marker of skeletal muscle cell. It is a marker of skeletal muscle cell. Then there are others also. G, glial fibrillary acidic protein. Glial cell, glial fibrillary acidic protein. This is a marker of astrocyte. So what you are finding out here, the bottom line is that intermediate filament is a fixed structure which is specific for a specific type of cell. Like for example, cytokeratin is specific for epithelium, vimentin is for mesenchymal, desmin is for skeletal muscle, and glial fibrillary acidic protein for astrocyte, and there are many more. So these are known as the intermediate filament, the cell marker or the fixed cell cytoskeleton, right? So this is all about your cell